we're back on at the right time. How do I do this? I feel like I've given a couple talks on Zoom, but every time I'm like a bit confused. Stop share. There we go.
Okay. Not the only one.
If I did more teaching these days, I'd probably be more comfortable on it. I still have a couple colleagues who ask to meet on Skype, which is what I'm most comfortable on.
I think I've used Skype in over a year.
Yeah, it's not a thing anymore. Actually, a couple people have. I've started using Google Meet. It's pretty easy, just from Gmail to send a link. Like it's faster than Zoom, actually. So that's
Plus,
kind of what
Microsoft has Teams now, which is their product, right? So maybe they're going to stop supporting Skype eventually and just abandon the product.
Yeah, that's what I figured. Absolutely, yeah. They're pushing it on universities too, I think, because they get it for free, probably with the Windows licenses and whatnot. I know a few students at UDM originally had pro licenses for Zoom, and that was all cut out. So they're putting everyone onto Teams now. Is the volume of the, like, is my mic good? Can you hear me well?
Yeah, seems
loud enough.
Yeah.
Okay, perfect. How do you do this usually? Do you have people post like questions in the chat? I'm happy for people to do that so that I can answer them at the end of the talk, or if people prefer to ask them in person, that's also.
Yeah, I think whatever, pretty much whatever people prefer.
Yeah. I mean, I always try to promote asking in person because it's nicer, but usually we have quite a few questions that come in over the chat, so we can allow you to try to help facilitate open communication, but it's our first seminar for the year, so we might be a little shy still.
That's fine. Yeah, I'm happy to take questions either straight from the chat, or if people feel like they want to have more of a discussion. Also, in person is great.
Yeah, I can keep monitoring the waiting room, but if we want to be timely, Graham, feel free to start and give a little introduction to our speaker today.
Great, thanks. Hi, folks. Thanks for tuning in. My name is Graham Raby. I'm an assistant professor here in the Department of Biology at Trent. And today it's my pleasure to welcome Dr. Dom Roche to our departmental seminar series. Dr. Roche is currently in year one of a 3-year Marie Curie postdoctoral fellowship, which is a very competitive postdoc fellowship awarded by the European Union. He's primarily based at Carleton University in Ottawa, but his award is co-hosted in Switzerland. Dom grew up in Montreal, and he did his undergrad and master's degrees at McGill University. And then he went to the Australian National University in Canberra to do his PhD, which he completed in 2014. And his PhD work focused on the behavior and ecophysiology of reef fishes on the Great Barrier Reef. And then Dom did a 4-year postdoc in Switzerland after his PhD in Redouane Bouchari's lab at the University of Neuchâtel, where he did more work on behavioral ecology and also other aspects of fish biology. The last couple of years, he's been based primarily at Carleton University, although he lives in Montreal. And Dom's not doing as much ecology work these days. He's shifted a bit more to focusing on doing research on open data, reproducibility, and other issues that basically involve improving how science is done. And so of his 50 or so publications, a good chunk of them are about issues around open data and reproducibility. And so I invited Dr. Roche because these topics on open data and meta science issues are really relevant to everybody in the department. So with that, I'll just stop talking and I'll invite Dom to take over.
Great. Thanks a lot for the intro, Graham. Yeah, I guess just to reiterate what Graham said, I have a background in ecology and evolution, but I've shifted recently to doing more meta science. So essentially the study, the science of science. And that's kind of what I'm going to be talking about today. So I'll just start sharing my screen here. Okay, can everyone see this properly? Yep, great. Okay, so yeah, the title of my talk is Why We Need Open Science. And you'll notice that it's a statement, it's not a question. And that's because as an early career researcher, I strongly believe that open science is the way of the future. And I hope that I'll be able to convince you of that in the next 45 minutes or so. So first of all, I realize that not everyone is necessarily familiar with the concept of open science. And I've divided my talk into 3 different sections for that reason. First, I want to tell you a little bit about what open science is and why it's important that we engage in open science. And then I want to spend some time telling you about how one goes about doing science in a more open and transparent way. And then finally, I'll talk about some of the work that I've been doing recently on the topic of open data and the challenges of getting people to share their data, as well as some of the solutions to that. So first, what is open science? There's a number of definitions that are floating out there in the literature and on the internet. This is one that I've put together that's quite brief. So open science can be considered practices that improve the transparency, credibility, and reuse potential of research results. Now, there's a number of different ways in which we can do this. It might include, for example, sharing your analysis code or script, sharing your data, making sure that the methods and the materials that you use are transparently and comprehensively reported. You can also do something called pre-registration. Many of you are probably not familiar with this. I'll explain it later in my talk. And then obviously a big component of open science is this the concept of open access publishing, right? I'm actually not going to talk about this in this presentation because it's probably the most well-known component of open science. And it also requires a talk of its own. So I'm going to focus on the other aspects that I've highlighted here. All right, so why do we need open science? And the answer is pretty straightforward. It's because we have a major problem in science right now. Many scientific disciplines currently face what people call a reproducibility crisis, whereby a very large proportion of research results can't be independently replicated. Now, the reproducibility crisis is also sometimes called the replication crisis. They're slightly different things, but they essentially point to the same problem. So replicability, so if I was asked to ask you to replicate a study, I would give you the data and the methods and probably the analysis script, and then ask you to replicate the results. So to see whether you can arrive to the same results using the data and the analysis methods. If I was to ask you to replicate the study, you would have to, I would give you a paper, explain how things were done, and then you would have to go out and redo the study on your own and see whether you come to more or less the same conclusion. So let me illustrate how serious this crisis is. In psychology and in the biomedical sciences, it's estimated that at least 2/3 of published findings are incorrect. There's a number of different studies that support this figure, and I'm just going to mention 2 as examples. So first of all, in a 2012 comment in the journal Nature, Begley and Ellis documented a really massive effort to replicate 53 landmark studies in cancer research. So these were really foundational studies that kind of led the way in the field of oncology. And surprisingly, they found that only 6 studies, so 11%, were successfully replicated. In a more recent paper published in 2015 in Science, there's a group of authors that calls itself the Open Science Collaboration that tried to estimate the reproducibility of psychological science. So they tried to replicate 100 studies in 3 psychology journals using high-powered designs, so large sample sizes, and also, whenever it was possible, using the original materials from the study they were trying to replicate. And the conclusion of this massive effort was that only 39% of the effects were rated to have replicated the original results. I don't know how you feel about this, but it makes me fairly nervous. I mean, we're talking here about fields that have a direct impact on the health and the well-being of humans. So if you're not convinced, let me read you a quote here from Richard Horton. He's the editor-in-chief of The Lancet, and The Lancet is probably the most respected and prestigious journal in the medical sciences. So in an editorial in 2015, Horton wrote, "The case against science is straightforward. Much of the scientific literature, perhaps half, may simply be untrue. Afflicted by studies with small sample sizes, tiny effects, invalid exploratory analyses, and flagrant conflicts of interest, together with an obsession for pursuing fashionable trends of dubious importance, science has taken a turn towards darkness." Now, I think we all agree that this is a fairly bleak and dramatic statement, but I think it carries a lot of weight coming from someone as respected as the editor-in-chief of The Lancet. Okay, so reproducibility is clearly a problem in psychology and in medicine, but how serious is it in our field, in ecology and evolution? And the answer to that question is, well, we don't really know. Why? Well, because there's only a handful of replication studies in ecology and evolution. And this was shown in 2019 by Clint Kelly, who's a researcher here in Montreal based out of UQAM. So he used text mining to scour through almost 40,000 open access papers across 160 journals in ecology and evolution. And he found that only 9 papers, so 0.02%, claimed to have replicated a previous study. Now, the limitation of the study is that Clint only looked at the open access literature because he was using text mining. And so I asked him, well, what do you think the replication rate is across the entire literature in ecology and evolution, closed and open access? And he estimates that the replication is far below 0.1%. So it means that there are very few studies that are repeated in order to see whether they actually hold true if they're done by another research team. So here's a good example of a recent high-profile replication study that wasn't included in Clint's analysis. Lots of papers over the last 3 decades have reported really strong effects of color bands used to mark zebra finches. So some colors, like red, you can see here, are used as textbook examples of how artificial color can change the attractiveness of males or females in this species, and therefore increase their reproductive success. In 2018, a group of authors conducted a study that had a total sample size of 730 finches, which is more than the combined sample size of all 23 papers that clearly supported the color band effect hypothesis. And the results of this replication attempt showed that color band doesn't explain any of the variance in attractiveness and male or female fitness. So here's another example from a study that Graham and I were involved in. We published this early last year, and it caused a bit of a stir in our field. I see Graham smiling there. So this was a 3-year replication study by 7 people looking at how end-of-century levels of ocean acidification affect fish behavior. We tested over 900 fishes across 6 different species and 2 life stages. And we looked at the 3 most common behaviors that had been looked at in previous studies examining the effect of ocean acidification on fish behavior. So we looked at avoidance of predator chemical cues. We looked at general activity levels of fish, and we also looked at something called behavioral lateralization, which is essentially the tendency of fish to turn left or right when they face a barrier. Don't ask me why people look at this, but it's very common in studies of fish behavior. So we filmed all of the behaviors that we measured. We used automated tracking software to extract the data and remove any potential observer bias. And we made all of our data and code publicly available. So anyone can go back and repeat our analyses and see whether they come to the same conclusions. And like the title says, we found no evidence that ocean acidification disrupts the normal behavior of these fishes. So what's really striking about this is that our results contradict a whole slew of high-profile studies reporting very serious effects of ocean's acidification on reef fish behavior, including that fish are attracted to the chemical cues of their predators in low pH. So hopefully this gives you a sense of some of the big issues that we're facing in science and in our field as well. And one thing we can argue, though, is that irreproducible results in ecology and evolution might not have the same consequences for human welfare as in medicine and psychology. But, and very importantly, I think, irreproducible results in our field can still have devastating effects on the public perception of scientists, and they can also promote distrust, mistrust, and denial on topics like evolution, climate change, overfishing, pollution, etc., etc. I could go on and show you more studies and opinion pieces that document the scale of the reproducibility crisis, but if I did that, I think we'd still be here tomorrow. So instead, it's interesting to ask the question, well, what does the average scientist think about this problem? Experts seem to think that it's a major issue, but what about the non-specialists, your everyday researcher? The journal Nature tried to answer this question by surveying over 1,500 researchers across a range of different fields. And the results were published in a 2016 editorial piece called Reality Check on Reproducibility. So these are the main results from this survey. There were differences, obviously, among fields, but overall, 52% of the surveyed researchers answered yes, I believe that there's a significant reproducibility crisis in my field. 38% said, yes, I think there's a slight crisis, whatever that means. And then, interestingly, 3% of the respondents were able to say confidently, no, I don't think there is a reproducibility crisis in science. And just out of interest, 3% is also in the ballpark estimate of how many people fabricate data in science. So you can make of this whatever you want. Researchers are not the only ones who are alarmed by this problem. The public is also taking notice. And this is pretty clear from articles published in magazines and newspapers, very well-known magazines and newspapers like The Atlantic, The Economist, and the New York Times. So because of this, the reproducibility crisis is also often referred to as a credibility crisis. Now, if you search for credibility crisis on Google, this is what pops up, obviously. But Donald Trump aside, it's critical that scientists avoid losing the public's confidence because at a time of post-truth politics, accelerating climate change, pandemics, and anti-vaccination movements, science can't really afford a credibility crisis. There's so many pressing societal issues that need solving right now, and science is a big part of the problem. But for science, or sorry, a big part of the answer, not the problem. So, but for science to be effective, it needs strong supporters, including politicians and obviously members of the public. Why do we have this crisis on our hands? And again, I think here the answer is fairly simple. We live at a time where the culture in academia can be described by three very famous words, which I'm sure you're all familiar with: publish or perish. Funding agencies and universities are putting a lot of pressure on researchers to churn out papers, preferably in highly prestigious journals. This leads to what some researchers call the natural selection of bad science. where selection for high output leads to poorer research methods and increasingly high false discovery rates. Essentially, results that don't hold up when we try to replicate or reproduce them. But you might wonder, well, why is it that researchers wanting to publish in prestigious journals use these poor research practices? And that's because often they lead to more exciting results that are easier to publish in these highly prestigious journals. What are these poor research practices? Or as Richard Horton, the editor of The Lancet, calls them, the bad research behaviors that contribute to generating these false positive results. There's a whole bunch of them, and they're very well explained in a 2016 paper here by Tim Parker and colleagues that you can find in Tree. I won't have time to cover them all, but I'll briefly mention a few that I think are particularly important. For starters, confirmation bias is the widespread human tendency to preferentially search for and interpret observations as consistent with one's belief about how the world works. This is problematic, obviously, because it means that researchers aren't impartial when they're investigating their research questions, and they'll tend to favor their pet hypotheses, either consciously or unconsciously. And the consequence of this is an increase in the rate of false discoveries. Selective reporting, which is also known as cherry picking, is the practice of reporting only a subset of the hypotheses tested. And anyone who's taken a first-year biostats class will know that the more hypotheses you test, the greater the likelihood of getting a significant result just by chance. So if you test 20 different hypotheses, but you only report the results that came out to be significant, the 1 or 2 results that came out to be significant, it's very likely that those are false positives. Another poor research practice that's on par with selective reporting is something called p-hacking. And I really like this cartoon because I think it says it all. If you torture the data long enough, they will confess. More formally, p-hacking is defined as a variety of practices that increase the odds of finding a statistically significant result. For example, by conducting multiple versions of an analysis with different covariates, interactions, or subsets of the data. And you do this until you find the result that you want, and that's what you present in the paper, and you fail to mention all the other statistical tests you did that led you nowhere. So again, the consequences of p-hacking is an increase in irreproducible results in the published literature. All right, so I've been talking a lot about all the different problems that are the root cause of the reproducibility crisis, but now I want to switch gears a little bit and talk about more positive things. How do we fix these issues? And for me, finding a meaningful solution to the reproducibility crisis requires a cultural shift towards more openness, transparency, and collaboration among researchers. This is essentially synonymous with open science. And this leads me to the second part of my talk, where I want to explain what does it mean to do open science in practical terms. So simply put, open science requires that we improve our research workflow. What's a research workflow? It's the sequence of activities and procedures at different stages of a research project. So earlier career researchers like myself often focus on the later stages of this research workflow because we're concerned about getting a job. And so we'll focus on getting exciting results and publishing them in a great journal. But we all know that there are many other steps that come before this, really important steps. For example, asking a scientifically important or interesting and valid question, and then developing hypotheses and predictions that flow from this question. Once that's done, we have to think carefully about how we're going to collect our data and analyze them to test these hypotheses. And one way that this can be done in a more rigorous and transparent fashion is through this thing I mentioned early on in the talk called pre-registration. So pre-registration isn't yet a mainstream concept in ecology and evolution, but it's required for things like clinical trials in medical studies. It's the process of making and registering key decisions about the study design and the data analysis early on in the research workflow before we go out and collect the data. So you can think of this as, for example, writing a research proposal for a grant or your qualifying exam, where you would carefully describe how you're going to collect the data and how you're going to analyze them. And then you would archive that proposal in a permanent database where it gets timestamped. And the point of a pre-registration is to show reviewers and readers, hey, I actually said I said that I was going to do the things that I did beforehand. I didn't cherry pick my results or p-hack my way into finding these really exciting results that I'm presenting in this manuscript. So this is how you would go about it in practice. You can pre-register by going to an archive, for example, the Open Science Framework, and you would simply create an account and then upload your registration for it to get a timestamp. So this is an example here from one of my pre-registrations. So this is a study that I did on data sharing by Canadian researchers. And you can see here that in 2019, I uploaded this document that describes how I'm going to go about this study. I chose to keep this document private for a year. You can do that, not that I was worried about it, but you could do it if you're worried that people are going to steal your ideas. If you wanted to, you can also release it publicly immediately. This is what the actual form looks like. So it's not, you would take your proposal and then use it to answer a bunch of questions that are asked of you. And then that's the actual document that gets timestamped. So another way to register a study, which is more intense than a simple pre-registration, they're not the same thing. It's via something called a registered report. So registered reports are a publication format where peer review occurs prior to data collection and analysis. So I'll try to explain it here with this, this little diagram here. So the same way that you would do pre-registration for a registered report, you come up with your ideas, you write a proposal basically, and then you submit that. Rather than putting it on a repository, you submit it to a journal. That proposal goes out for review. It comes back to you with comments from the reviewers. If it needs to be improved, you can improve it, resubmit it, and then just like a regular manuscript until the reviewers accept it. Once it's accepted, you go out, you collect your data, analyze them, you write your manuscript, and then there's a second stage of the review where the final manuscript gets sent back out for review to the same reviewers from stage one, who essentially assess whether you followed through with your initial registered methodology. But what's really interesting about this publication format is that accepted papers are virtually guaranteed publication in the journal if the authors follow through with the registered methodology. So that's really fantastic because it means that if you have a good idea and your methods are sound, your paper gets accepted, irrespective of what the results are. So there's no pressure for you to come up with really fantastic results. You just have to follow through with what you said you were going to do. And just in case you're wondering, I checked this morning how many journals actually accept this new type of publication. And currently there's 275 of them listed on the website for the Center for Open Science. And many of those are journals in ecology and evolution, or at least general journals that would accept work in our field. One question that people often have is, does pre-registration allow additional data collection and analyses? And the answer to that is yes. Non-pre-registered measurements and analyses are allowed, but they clearly have to be labeled as exploratory instead of confirmatory in the publication. And that's because people tend to have more confidence in confirmatory rather than exploratory results. So although it seems quite rigid at first, pre-registration is actually quite a flexible tool for helping us reduce the prevalence of some of the poor research practices that I mentioned earlier on, especially things like selective reporting and p-hacking. Now, the third step in our research workflow is data collection. So improving the step can be done by ensuring that we're blind or unaware of the experimental treatments when we collect the data, and by carefully documenting the data collection process so that our methods are sufficiently described that anyone else could repeat the study without our help if they wanted to. Blinding is something that most of us are probably familiar with, but we often fail to implement it in our studies. So the classic example is the medical single blind study where subjects are unaware of the treatment that they're receiving. But clearly that's no longer allowed because we know that the doctors themselves are subject to their own biases. Obviously, for those of us who work on plants and animals, studies are always single blind, but we should really work hard to make them double blind to reduce these conscious or unconscious biases that could affect our results. And that's really important because a 2015 study by Luke Holman and colleagues found that blind protocols are not only very uncommon in the life sciences, but also non-blind studies report higher effect sizes and more significant p-values than blind studies. So beyond blinding, carefully describing the materials and methods that are used is also key to allow others to understand what was done and also eventually for someone to replicate the study. I think this cartoon speaks for itself. Here in this PhD comic, the evil professor says, no, if you make the paper too easy to read, everyone will know how you got the results. But we all know that that's the purpose of a materials and methods section in the first place. So blinding, when it's well implemented, is really effective to reduce confirmation bias, and therefore it contributes to making our workflow much more rigorous and credible. Providing a complete and transparent description of the materials and methods also increases the credibility of our results because it allows independent replications. So the last stages of the research workflow I want to mention are data management, so organizing, parsing, and cleaning data, and the statistical analyses, preferably using code or script-based software rather than a software where you click and drag. One way to do this in a more transparent way is via open data and open code. For the rest of my talk, I'm going to focus on open data because that's what I study these days, but it doesn't mean that open code is not as important. It absolutely is. So what are open data, or what people often refer to as data sharing? It's the process of making data and associated metadata to replicate a study's results publicly accessible in an online repository. Metadata are data descriptors, so information about the data set to allow people to understand what the data are and what they mean. There's a number of different data repositories or databases that we use in ecology and evolution to archive and share data. I've put a couple examples down here. You might be familiar actually with these repositories because you would have come across them if you've recently published in a journal that requires data sharing as a condition of publication. So these are just some examples of journals with a strong data policy. So they require data sharing as a condition of publication. And the list is much longer than that actually, and it's growing every day. So why are journals suddenly deciding to implement these policies? The reason is because there's a lot of benefits to data share, especially for the scientific community and for the broader society. So open data are really good because, for example, they avoid data being lost to science, either because CDs become obsolete, hard drives can be broken, and also researchers move on in their careers. They might leave academia or they might retire or die and not pass on their data to other people. So the data are lost to science. Open data also accelerates scientific discoveries by allowing researchers to combine data and answer potentially bigger and broader questions. They improve our return per taxpayer research dollar. And this is important because our grants and our salaries as researchers are paid for with public money. Open data increase our ability to reproduce and validate results, as we've been discussing, and they also facilitate detecting fraud whenever it happens. So if you've been following science, the science news last year, you probably would have heard of Jonathan Pruitt at McMaster. I think he's got close to 10 retractions now and dozens of papers that are being looked at. And if it hadn't been for journals requiring open data, this fraud probably would have never been identified. Okay, so despite these obvious benefits for society, it's not really easy to get researchers to share their data, at least not all the time. And for the time that I have left, I'm going to focus on some of the challenges of getting people to share, and also some of the solutions to increase participation in open data, including some of the research that I've done on the topic. In theory, open data is really great. You publish your study in a journal, you post your data in a public archive, science progresses, and everyone's happy. In practice, that's not really how things are playing out. A lot of researchers are very reluctant to make their data publicly available, even when they're required to do so by the journal where they publish. And this pushback seems to be particularly strong in ecology and evolution, perhaps because data sets are often complex, they have a long shelf life, and they can also be used to test multiple hypotheses. So researchers can reuse their data to publish more papers. Some of the reasons for why people are reluctant to share their data include the fact that proper data archiving takes time, and this is time away from publishing. And right now, what's rewarded in academia is publishing, not data sharing. There's a lot of competition for publications, and so researchers can be worried that they might get scooped if they share data for a paper, but they intend to reuse these data for another study. Some researchers also have concerns about errors being identified in their work. That's understandable. We all make errors. However, you could argue that error detection is good for the scientific community and society, but obviously comes potentially at a cost for the researcher. Some people are also concerned about others misinterpreting or misusing data that they're not familiar with. And finally, there's just a general lack of recognition for open data. So these are what I would think of as perceived fears. These are things that people mention when you talk to them or in surveys about their concerns about open data and what they worry about if they were to share. A student and I recently conducted a survey in Canada. We contacted 365 profs from Canada's top 20 ranked universities, and we got answers back from 140 of them. And we asked them about whether they had actually experienced any of these costs. And again, these are self-reported answers, so they might be an overestimate, but overall, you can see that, on the y-axis here, this is the percentage of respondents, and then on the x-axis, you have the different types of costs. So misuse of data, being scooped, not being offered co-authorship on a paper that you felt you should have been a co-author, et cetera, et cetera. This last one here is public shaming from errors being identified in your work. And overall, you can see that it's actually not that big a deal. And the category that has the biggest number of people that experience this is misuse of data. And that's still 10%, only 10% of people. So given this pushback that I just mentioned, it's interesting to see, well, what the data sharing trends look like in ecology and evolution. And thanks to the work of Tim Vines and others, we know that these mandatory open data policies have had a very positive effect on data deposition rates. So more people are depositing their data in these repositories. We have more open data. But it's worth pausing here for a second and asking, well, does more actually mean better? Especially since most journals and databases don't actually verify the quality of the shared data sets beyond basic things like ensuring that there's a data availability statement in the paper and that the files that are archived are free of viruses. So I was interested in looking a bit deeper into this question and teamed up with some colleagues from the Australian National University to look at the quality of the data that we're sharing in ecology and evolution. And this led to a paper in 2015 in the journal PLOS Biology. So we did this by assessing the data sets associated with 100 non-molecular ecology and evolution studies published in journals that had a mandatory open data policy. So we looked at 50 studies here in 2012 and data sets from 50 studies in 2013. And we assessed the quality of these data sets based on two different criteria. First, their completeness. So were all the data necessary to reproduce the results available in the data set? And secondly, we looked at reusability. This is not required. It's not part of the policies of these journals, but it's still very important. Reusability has to do with how easy is it for other people to understand and reuse the archived data. Are there sufficient metadata to describe them? Is the format used by the authors, does it facilitate reuse, or does it make it really hard for other people to try to use the data? And we ranked data completeness on a scale from 1 to 5. A score of 4 or 5 meant that the data set met the open data policy requirements of the journals, of the different journals, whereas a score of 3 or lower meant that the data set didn't actually meet the policy requirements. And we did exactly the same thing with reusability. So 4 and 5 were deemed to be accessible, and 3 and below were deemed a fail. So here are the results. First, for data completeness, here you can see the number of studies on the y-axis and then the score that they got for completeness. And again, a 4 above was a pass, 3 below was a fail. And essentially we found that more than half, so 56% of the studies didn't meet the minimum requirement of their journal's open data policy. And what about reusability? Well, here we found that even more, so 64% of the studies were archived in a way that either partially or entirely prevented their reuse. So I think it's possible to see these results through 2 different lenses. On one hand, it's fairly sobering to see that the majority of what's being shared out there is unusable. But on the other hand, it's encouraging to see that a bit more than 2/3 of these studies shared pretty high quality data, especially given that open data wasn't really common back in 2012 and 13. Still, this begs the question, well, how do we increase high quality participation in data sharing? There's 2 approaches that we can use for this. First of all, we can use the stick method and impose more constraints and requirements on people, or we can give them a carrot and reward good behavior. And I've argued in the past that we need a healthy mix of both. But unfortunately, right now, journals, funders, and universities are really prioritizing the stick method over the carrot. Or at least that's been my experience. So what's come of that? Has this strategy led to an improvement in the quality of open data over time in ecology and evolution? This is something that I looked at recently by using exactly the same methods as I showed you before and checking the completeness and reusability of 25 data sets per year from 2012 to 2020 that were associated with studies published in journals that require data sharing. So these graphs here show data completeness and data reusability for 25 studies per year across all these different years. And the points that you can see here were a random sample of the data set I collected for 2012 and 2013. And you can see that there's no obvious difference between those 2 years. What happens when we add another 7 years of data? Unfortunately, absolutely nothing. So not the slightest sign of improvement. I was really surprised and disappointed to see this. But maybe this is a sign that we need to rethink our strategy to get people to share better quality open data. So very briefly, I just want to show a couple more results. So this is fairly disappointing, but what next? What can we do with these data? So we can at least try to understand something about who shares well and who doesn't. In other words, what are the factors that might explain the variance that we observe in the quality of these data sets? One possibility that's been brought up is that nice people are the ones that share higher quality data. In fact, data sharing is considered by many people as an altruistic behavior. And that's because currently the individuals or the individual benefits of sharing data are fairly limited, whereas there's lots of perceived costs. So one possibility to explain differences among researchers might be that data that are more complete and more reusable are shared mostly by researchers who tend to be more cooperative in their everyday life, in contexts that are unrelated to science and data sharing. And I teamed up with psychologists in Switzerland and the UK to try to answer this question using a subset of the data that I just showed you. And we used a survey and an online experiment to measure each researcher's level of cooperativeness and then relate their cooperativeness score to the quality of their open data sets. The full analyses are fairly complex. We used a multivariate Bayesian model. So what I'm only presenting here is a simplified version to save some time. But lo and behold, we found no difference whatsoever. Researchers who are highly cooperative shared data sets that were no more complete than the data sets of non-cooperative researchers. And the same was also true for data reusability. So lots of negative results. So what does this mean? It means that nice people can't share, or at least that cooperative researchers aren't any better at sharing their data than researchers who act less cooperatively and report themselves as being less altruistic. Clearly, some people don't want to share, and that needs to be addressed through cultural changes and the right set of incentives. But for those researchers who are cooperative and do want to share, the fix is pretty easy. We need better training in data management. It's hard to share when you haven't really been shown how to do it properly. So luckily, universities are starting to understand this, and there's lots of initiatives that already exist to teach scientists how to be better at managing and sharing their data. For example, data carpentry and Foster are really well-known initiatives, but there's many more out there. And I think that thanks to these efforts, the future of open data is going to look a lot brighter, hopefully much soon or very soon. All right, so I'm going over time here. I need to wrap this up. If there's one take-home message from this talk, it should be that curbing the reproducibility crisis requires that we make considerable improvements to the way that we do research, our research workflow. An excellent way to do this is by adopting more transparent, open science practices that increase the credibility, the reproducibility, and the reusability of research results, while also accelerating scientific discoveries. There's obviously challenges to implementing these different measures, but all of them have pretty easy fixes. And we've seen a lot of progress in the last couple of years with many funding agencies, journals, and researchers getting on board the open science movement. So finally, if you're interested in this topic, you can read up on the reproducibility crisis in a really fascinating book published a few months ago by the psychologist Stuart Ritchie. And you can also get involved in the brand new Society for Open, Reliable, and Transparent Ecology and Evolutionary Biology that we launched last December. So check out the website or the Twitter handle if you want more information. And with that, thanks very much. Apologies for speaking too much. I tend to do that, and I'd be happy to take questions.
Thanks, Dom, for the excellent talk. I'll, I guess, host the questions and stuff until 2 o'clock, and then I'm going to be getting a phone call around then, so I might just disappear suddenly. If that happens, Megan, I'll let you take over. But we have some questions, I guess, in the chat and comments that we'll go through first. So Maggie Xenopoulos, who's an ecologist in our department, Dom, she had a few comments. I guess I'll read her comments first, and then there's a question as well. So the first comment was, as a culture change may be needed. As an editor-in-chief myself, I can attest that most peer reviewers do not think that repeating studies is novel, thus recommending rejection of the paper. Journals like PLOS ONE may be changing this. Second comment is that often the methods do not have sufficient detail to truly reproduce studies, especially for short format journals like Nature. So I'll let you talk about those ones.
Yeah. So to the first point, can you remind me quickly what the first point was? I had the second one in mind here, but
it's the issue of basically peer reviewers suggesting that papers should be rejected if they're just repeating previous studies and that they're not novel enough for the journal.
Yeah, I think that is obviously, part of the cultural change that's needed. That being said, I mean, there was the mention of clause 1, potentially like, incentivizing or at least accepting kind of replication studies. I would say that that's actually changing. It has changed already. I mean, just the two examples of the replication studies that I showed you, one was in ecology and the other one was in Nature. Not only were they replications, they were negative results as well. I can give you a bunch of other examples. I mean, there's so many examples, not necessarily in ecology and evolution, but other fields that are related of replication studies published, for example, in eLife. It's also an open access journal, but it's highly prestigious. Peer review is pretty intense. It's hard to get in. PLOS Biology also regularly publishes replications, and they actually put out calls for them as well. I'm aware of a special issue for a behavior journal. It's not animal behavior, it's another one that was specifically focused on replication. So the whole issue is just going to be that. So even though, yes, that was the case, and it might still be the case in some journals, I think that things are changing right now.
You can probably stop sharing your screen, by the way, Dom.
I thought I had. Oh, sorry.
We'll,
yeah.
The
Sorry
question
about that.
from yeah, that's good. The question that Maggie had was in terms of the open data stuff you were talking about, as far as how well people are doing with sharing their data properly and having data sets that are properly archived. Whose responsibility is it to ensure compliance with these policies? Is the editors, the funders, or the authors in terms of making sure people are sharing data properly?
Right now, everyone's and no one's basically. The journals have this policy, and then, I mean, I've seen some journals that are happy with statements that saying like all the data are in the manuscript, and obviously that can't be the case, right? I mean, unless you have massive tables, depends on your study, obviously, but what we've seen is that a lot of things are slipping through, in part because editors and reviewers are overworked. I've spoken with editors and they say, I don't even check that. Even though the journal has a policy, they don't look at it. It's obviously the author's responsibility. I think part of it is your reputation also hinges on whether you do things correctly or not. So from my point of view, I would want to share things properly, because if people start looking at it, you want to show that you were doing things correctly. And I think it comes back to this point I made in the talk that achieving high quality open data or good sharing practices has to be a healthy mix of requirements and incentives, right? It's impossible to police everything, even if reviewers were told to check very carefully. And some journals now are even starting to have like data editors. So people that are going to look at the data. People who don't want to share will find ways not to share. It's possible. Some data sets are really complex. There's all sorts of things you can do with data to make them hard to reuse. And so I think that we need to, although we need to have these policies in place to get people to realize that it's important, we also need to reward people for doing things well, right? So for example, funding can be dependent on the stuff that you're sharing basically, and how often things are reused, because we need people to do it out of their own accord. We can't force them to do things well. I think that would be really difficult it wouldn't necessarily be a successful strategy, I think.
So the next question in the chat there is from Samantha Collins, and it's similar to what you already talked about do you think there needs to be a shift in ideology and publishing practice to reduce publication bias and it's often hard to publish studies that are not considered novel.
Mhm.
And Samantha also says, great job on your talk. But I think you sort of talked about that already, so maybe that's because that's pretty similar to Maggie's questions.
Yeah, thanks. I mean, what I can reiterate on that point is also like that's the beauty of these registered reports that I mentioned. People are often confused by like what's a pre-registration and what's a registered report. I hope it was clear in my talk that they're not the same thing. But if you're a student, for example, you have a brilliant idea or a PI and you really want to do the study, you think it's going to be great, but obviously we're all nervous about what we're going to find, right? I mean, most often, as you can tell from the results I've shown you, I end up with non-significant results. But it doesn't mean that my ideas are bad or the way that I did things is bad. And so I find this new publication format really exciting because it can really relieve that anxiety, I think, or the stress of having to find exciting results. You can actually submit your proposal to a journal and it'll get accepted before you do the study, right? So that's a nice way to kind of, you have to think of a novel. It has to be interesting. It has to be a novel study, quote unquote, but at least you don't have to worry about what you're going to find.
Cool. Yeah, I agree with that. So the next question on the chat is, what do you say to critics who claim that open science inhibits science and innovation by removing incentives to do research?
So I guess that the question relates to like IP potentially and things like that. It's interesting to see. So I tend to go and explore what's happening in fields other than ecology and evolution. And I've interacted quite a bit with people that are in neuroscience, especially here in Montreal. There's the Montreal Neurological Institute. They're developing drugs and therapies to treat neurological diseases. And you would think that like they would want to stay as far back as they could from open science because there's a lot of money to be made in these treatments. But in fact, that's not the case at all. They are one of the institutes across the world that have really embraced open science practices, and they've been so much more productive for it also. And even though there still is like, there's the conflict of like intellectual property versus sharing, they're able to navigate that quite well. Their partners haven't shunned them, basically. They're getting more funding as well from funders that want the research to progress faster, right? I mean, I think everyone will agree that if we share, society benefits, right? Keeping things to ourselves tends to be, benefit the individuals or the societies that hold the knowledge. So for science, I think the community generally agrees that open practices are good. And I think there are a couple examples out there, the neuro being one of them, showing that in fact, you can still, get funding, be supported, when you do open science. And one of the interesting things that is discussed in the medical sciences is also the demand coming from the patients, right? You have to recruit participants to be in these medical studies. And now, often patients actually refuse to participate unless their data, their anonymized data, right, without any identifiers, is shared to help science progress faster, because a lot of these people have like terrible diseases, might die in a couple of years, and they want to find a cure faster. And they understand that if we share, we will come to that cure faster.
Another question came in from Rachel saying, as everyone here knows, sometimes experiments fail or methods have to be drastically changed due to unforeseen obstacles. How would those situations fit in a pre-registered science framework? And thanks for the great talk.
Thanks. That's a really great question. I had a slide about it, but I cut it out, thankfully, because I did go over time. So that's one of the concerns that people have with pre-registration. Like, it's impossible to think of all the things that are going to go wrong and the ways in which your experiments might have to change or observational studies. example, I mean, I go out in the field, I still work on fish, basically. I go out in the field, plan to work on this particular species and do stuff, but actually there's not enough individuals. That year, for some reason, like that species, I just can't find it. But there's another one I could work on. Does that mean I have to scrap all of my pre-registration? No. There, you can make amendments to your pre-registration. That is a possibility. And also, what people do often is that they will include a section in their paper. It can often be in the supplementary material, because otherwise it can be quite tedious to read, but where you say, well, here are the deviations from my pre-registered methodology, right? And I think people and reviewers are fairly understanding of these circumstances, especially for people doing field work or experiments where things go wrong. The purpose of the pre-registration is really to try and be as transparent as possible. And it's for the readers to have, to increase the credibility, basically, of the research that you're presenting. And in being transparent and reporting those differences, people, I think, recognize that. And in my experience, it hasn't caused any problems with publishing pre-registered studies.
So, yeah, Peter, go ahead.
So I just had a question about like what you think, how much we can improve upon the reproducibility crisis. So you presented some crazy staggering numbers there at the beginning about, the ability for reproducibility in our field. And I'm wondering, like with pre-registration being enforced and data sharing being enforced, and all of these methods that you talked about being enforced, how much of it do you think is kind of just embedded in the ignorance of the experimental design, things that we can't foresee, things that we can't control, variables that come into play with regards to those reproducibility numbers? Like, I'm just wondering how much you think is actually that we can affect and make change